I mean, it was a very different. I mean, we have seen the problem of COVID-19, SARS-CoV-2 from different perspectives. I think it was a nice gather of people doing different things, right? And trying to, the angles are very different from one or the other. So now the question is how can scientists help? Because there are many problems, right? And this is our just only a few problems that were mentioned today on how can we also collaborate more with virologists and material scientists, and also how can we solve these high societal problems, right? So how can we help, for instance, detection of these viruses? I mean, the problem of detection is being, at least in the US, it's been a problem because they take, in many places, it takes 10 days to get a result. So, and yeah, so in 10 days, it's too late, right? For some patients. So what can we do for this detection, for improving the detection? And is it possible also to work on these developments for antiviral self-sanitizing surfaces? So maybe I would like to open that to some, I mean, to the speakers, if they want to just comment on that, and then from there we can just let people discuss, right? So I would like to, if yeah, any of you of the speakers want to say something, then we can start from there. So there are two questions: is how can we help, and can we produce other types of materials for antiviral or anti self-sanitizing surfaces? So Suresh, I think you want to.
Yeah. So Mauricio, I think this is a very fascinating question. So I just wanted to make a comment. So I guess before we really address what we can do as scientists to help the testing, for example, let us take a step back and understand what the problem is. So if we rewind and look at some of the recent epidemics, so we talk about Zika, Ebola, and many others before, and if you scan the literature, there are so many novel sensing technologies, microfluidic devices, and all sorts of different things are published. So scientists are coming up with novel ideas, and there are certainly not a shortage of new ideas and technologies that, at least in the literature. But then come to practice, in 2020, we face a global pandemic, and there is a global crisis of testing shortages. And simply because despite the scientific advancement, the practical testing solutions are still the same probably a decade ago. So, and one of the important aspects is that the regulations of the testing, or in a diagnostic context. So, for example, in order to make a valid clinical diagnosis in the US for any disease, the test has to be done by a CLIA lab, which is a high complexity medical diagnostic lab, and the test must be approved by the FDA for that purpose. So the system is very rigid. And even now, when we are in the face of pandemic and everybody recognizes that testing is the most important challenge, and we need to come up with solutions, there are several emergency use authorizations being given by FDA, but it's simply not enough. I think the first question is that we need to really come up with a framework, and this requires not just the scientists, but the policymakers and the regulatory agencies to really understand how can we make this more seamless so that the technological innovation can be implemented into a clinical decision making. First point. And second thing is about the approach of being proactive versus reactive. So until the virus has emerged in China and several months into it, a test was not in fact available. So we are developing or creating a test after the virus emerged, and it is really spreading like a wildfire across the world. And then we started making a test. So the CDC test was several weeks after the pandemic was well underway. And then the problem is now, so you are trying to create or scale up the testing or testing kits in the face of a pandemic, which is simply not very practical way of doing it. So one of the key questions for this group and other scientists is, how can we be more proactive and come up with some of the examples that you shared are not really determined by the virus evolution of the virus type. So how can we harness this technological innovation of making more versatile and adaptable plug-and-play type devices that can be quickly implemented if, for example, in 5 years' time, 10 years' time, a new pandemic comes, and then potentially we might be in the same situation. So somebody has to design a RT-PCR assay, and then somebody has to start manufacturing them. So a key question is to two levels: how the regulatory agencies can then work with scientists and policymakers to make this seamless. And second thing is, how can we be more proactive and not really reactive, which is currently the problem. I'll stop there.
I think these are excellent points. And I think that's been always the problem, right? This is a regulatory agency. So you really want to have something in the market for testing. It needs to be approved, and it's a very tough approval, at least in the US, right? I mean in Europe as well, I suppose. But I think this is something that needs to be moving forward, and that goes into also the politics, and which maybe we should avoid talking about it. But and I think that's so. In terms of these regulatory agencies, do you have some ideas on how to? I mean, I'm sure this has happened to you several times, right? So how can we speed up processes? And I think they have some sort of fast track thing, approvals, but I'm not sure.
So there are some avenues, Mauricio. So for example, FDA has been fairly, I think, efficient this time around. They had really speed up the process of emergency use authorization. In some instances, people have received EUA in as short as few days. But still, the other issue is that, let us say we develop this graphene or whatever technology, and some of these technologies, they are only valid if they're used in a high complexity clinical disease lab in order to make a clinical diagnosis. So if I develop something and I can't really apply for an EUA because it's not valid, it has to go through a CLIA lab. And what really surprised me is that several CLIA labs, meaning the medical diagnostic labs that I have been interacting with, they are miles behind a decent molecular biology lab in any university. So some labs do not even have the basic hardware for molecular PCR, let alone something advanced. So there is this disconnect between the scientific advancement of the technology we talk about, but then the diagnostic practices are very conventional, and they're very rigid. So one of the options is that what we are talking now is getting an approval of FDA for a disease in the face of an epidemic or a pandemic. But there could also be another approach that some of these technologies, and if you see the literature, most scientists would publish a paper and it will just remain in a paper. We talk about, so if I have to give a talk about diagnostic technologies, I do a literature review and I say, oh, carbon nanoparticles or this, that, and the other. I'll have a whole group of sensors or technologies that I can summarize. But none of them would ever actually cross the scientific community and maybe amongst the people who review scientific literature and to actually become in practice. So that's where I think we can now focus more on not just the discovery, but how can we translate them, the translational potential, working with the industry, and maybe making some of these go through the FDA pipeline before the next epidemic happens. So if you have these, the reason PCR is so versatile is because it has been tested and it was used for several diseases. And then there is a high level of confidence in the medical community that this could be used. But what we do lack is that all these novel technologies, unless I understand the science, it's very hard to convince people that it works as well, or it works much better than the conventional tests. So we need to build that translational piece, which is missing. And once you have that, it's much easier to get the approval and probably then convince the medical labs to start using them.
Richard, can I make a comment here? I think you are raising a very interesting viewpoint. But as an outsider to this area, but I have dealt with developing sensors for the US Army and private industry for more than 2 decades. What surprises me here is even an approved technology like the RT-PCR. If you really look at the receiver operating characteristics, as I say, looking at false positives, etc., it's not really great. At least from the published literature on this COVID-19 situation, the numbers vary all over the place, and that is one issue, in terms of testing. My question is, after so many viruses, so many pandemics, why is that we haven't developed a simple point of care sensing? We still rely on something very sophisticated like the RT-PCR, which costs tens of thousands of dollars. Is there a reason? Is it purely regulatory, or is it something else that has stopped us?
Great question, Dr. Swaminathan. I think part of the answer to the question is in your comment itself. So let me try to give my perspective of how I understand this. So the first point you mentioned is about the diagnostic sensitivity specificity aspects, which will lead into false positives and false negatives. And one thing that we must keep in mind is that I'm a diagnostic clinician, so I understand this space. When you talk about a diagnostic sensitivity or specificity of an assay, it is always in the context of the prevalence. So, for example, if I have an assay that is the diagnostic sensitivity is 95%, it is not the same in a different place where the prevalence is 30% or 2%. And one of the key challenges currently we have is that nobody really knows what the prevalence of COVID-19 in any part of the world. So all of these estimates about the test being not very sensitive or specific are not really based on solid scientific grounding, first point. And second challenge is that truly, if you want to estimate the diagnostic sensitivity of an assay, you must always have a gold standard, right? So say I have a PCR assay, and there are 100 people who are confirmed positive by a gold standard assay, and then how many of those 100 my PCR is able to detect? And that's when you calculate the diagnostic sensitivity. But the challenge is, what is the gold standard for COVID-19? So when you say people are coming up with false positives, false negatives, what was the additional confirmatory test that is used to conclude that this much is false negative or false positive? And one of the problems is that then, if you compare a chest CT or X-ray with PCR, they are two different things. You're almost comparing oranges with apples, because what is one test is looking for as a clinical manifestation of the disease in the lungs, and the RT-PCR is looking for the virus. And the other complication with this one is that the timing when the test was deployed. So for example, as I think in your talk, you outlined very nicely, when a person is infected with the virus, there is a certain period that they become or remain virus positive. So if a sample was tested in that window for a PCR, you will be able to detect the person to be positive, because that is the time they're actively shedding the virus in their saliva or in the nasopharyngeal secretions. But if you delay by a couple of days, then the amount of virus shed goes below the threshold. So they are technically negative by PCR, but they're still positive. And then the next best thing you can do is then look for the signature in the body, which is antibody. So part of the problem is that since we do not know when somebody is being infected, you don't really know, is this a person a right subject for a RT-PCR assay or a serological assay? And now the other challenge is that the serology assays are not still being widely used. And most of the assays that are being used are also qualitative. So they will just say you have it or you don't have it. And based on our own research in Penn State, we now know, unless the serology assay is very well designed to a specific part of the spike protein called RBD, it cross-reacts with other endemic coronaviruses that you have mentioned. So if people are just using spike protein to create an ELISA that is qualitative, then your positive is not necessarily a true positive, because it could cross-react with other antibodies. So these are some of the aspects that really complicate the the assay sensitivity. And now the question about why we are not going past beyond this rather complicated and very cost heavy PCR and not alternatives. So if you understand the viruses, for example, there are multiple families of them. And just in Coronaviridae family, there are hundreds of coronaviruses that also exist naturally in animals, and hence when they shed their feces or whatever, it is in the environment. And the differences between them are not very significant. So it's not like a bacterium versus a virus. They are very, very similar. And hence, in order to make a very, very definitive diagnosis, you need to really capture the subtle difference between the viruses. And PCR has been tested to effectively capture that very subtle genetic difference between the virus. And they also have extremely low detection threshold, right? So they can almost detect close to 1 or 10 copies. So unless we really come up with an alternative that can provide the same level of precision and the sensitivity, analytical sensitivity of detecting a very low level analyte and still create the specificity of not cross-reacting with other closely related viruses or other entities, then you can't really beat the PCRS. And that's where we are stuck currently. So the technology has not really been able to address these 2 points, and hence still people are heavily reliant on PCR.
Okay. Oh, good.
Speaking of detection method, right? So I have a question to Mark. So Mark, from industry level, we know Collins Aerospace is heavily involved with our customer, the airline, Airbus, Boeing, for example. So from the point of view, from your side, right? What the industry view about those technology? Do we get any kind of suggestion from our customers? Say, hey, this is still not very mature. We need to work with the university, national lab, for example, right? Do we see those kind of very urgent requirement? What kind of view we have from industry?
So I'm not sure I follow the question exactly, but I'll try. They don't question if we're ready or if we're mature. They're not at the scientist end, they're at the other end, and they're ready to take action. And they really look to us to say, can you provide me antimicrobial material for my seats? Yes, I can put copper on your seats. Okay, how would that work and why? And let's go. So they don't really question the maturity. They basically leave it to us to make the case one way or another.
So that's I see. I'm talking about some detection, right, and the method. Things we're talking about that.
Okay.
Yeah. Do we see or have we saw, from look at the technology in US, for example, have we reviewed all this available technology? Say, here is a method that will give us a very quick detection about the virus, right? Either in the airport or inside the airplane, for example.
Yeah, so that piqued my interest a lot in that everyone agrees the number one thing to do is contain it and don't let it get very far into the journey. So don't let it get to baggage check or don't let it get to security. And so if there was a test where upon arrival, you could do whichever these test mechanisms you want to refer to, and it would be very quick to say, yeah, you're cleared. They are very eager to do that.
So from aerospace industry, it seems like we still haven't found anything. We're talking about quick detection. This kind of technology is not available right now. Is that right? Can I say that?
I'm not aware of any, and they haven't deployed any. Right now, we're talking about temp screening out of its simplicity, because another thing they don't want to do is actually go create a social distancing dilemma by creating lines and queues to get screened in. and basically bunching everybody up together in the first place. So they want to be able to collect the sample and then have them say, no, you're not healthy enough to go on a trip. The temp screening is an indication, but a lot of people, or the data I've read, are its accuracy and its usefulness is low percentages. And so between people being asymptomatic and what's the reason they have a fever, there's a lot of people looking for, are there other simple, quick things like checking your respiratory rate that you could also do very quickly and say, okay, you're at least a candidate. So if your rate was off, your heart rate, your respiratory rate, your temperature, we're going to pull you aside, and then with the idea maybe then there's a better, more purposeful test that could be done to confirm or deny, and everybody that didn't pass that screening could just keep going on their journey.
Yeah, I think that's the area, I think, the industry and academia can work together, right? If we're talking about, all this opportunity, that's really one of the key area we need to work out as soon as possible.
Yeah, so they would be very eager to do that as soon as possible. You'd be reading it in every news article in the globe if you had something like that quickly. At the other end, for the airplanes, it's more of a long-term thought to say, so as we are the virus movers, because we move the people that have the virus, should we actually put a sensor in the airplane? Because then you would have a sample, you'd have a manifest, who it was, where they came from, where they're going, and you could start detecting it and doing contact tracing or trends and go, wait a minute, we got a new one. Okay, we've had 3 flights of that same signature of a test result come out of that same airport. So should we put a pause on travel from that particular airport for a week instead of not knowing and letting it run the globe for a month and then go, okay, now we have no choice but to shut down the whole planet?
Yeah, I think that I would suggest there might be a really good opportunity. Let's work together from the industry and academia in the virus detection area, right? Maybe can get some funding from the government. We may also get some match funding from the industry, from the company. Yeah, let's think about that.
And then there's the logistics and practicality side. In my company, we always talk about the use case and walking through it. So say you did have a test positive result for something that was new or novel. So now what do? You don't know what it is yet. You have somebody on the airplane had it. You don't know if you now have an airplane full of it. Do you quarantine the airplane? What are the next 3 steps after that then for the people that are now living in this situation? It's hard to think about where you go with that. I agree with you, though, we should go, and we should get some more people to help solve that problem of, assuming it does test positive, what do you do in the immediate term with that plane load of people?
So that is to me, it's kind of go to another topic, right? So for this workshop, at the very beginning, we try to cover 3 areas. One is how to use the cover material to trap the virus, right? The second one is how to detect the virus. Now we need to detect the virus as quick as we can. So that's the second one we just talked about. The third one is really the virus inactivation. I think, Mark, you talked about what we need to do next is more related to the virus inactivation. Is that right?
Yes. Of course, we'd want to deactivate it, but if it's new, we might not yet know how.
Yeah.
That's true.
So then it's like, we even talked about, do you have these shipping containers that are essentially portable negative pressure isolation pods? And do you go take those somewhere and put them there and say, okay, you guys all have to be quarantined for a little bit while we figure this out?
I think, I mean, just trying to combine the first part of the discussion and the second part of the discussion, I think we need to understand everyone's needs, right? So what are the technological needs from industry at different levels, and what are the current problems that you have that need to be solved? And at the same time, we need to also, what Suresh was mentioning, what are the problems we can solve, and what are the questions we need to answer from the fundamental point of view, right? So that we can collaborate, we have one common objective, industry, scientists, engineers, and even and regulatory agencies at some point, right? So how can we play into the same field? So we need to find things in common, and we want to have the same objective, so we just can concentrate on one application, one thing, or the other application and the other thing. And I think trying to have this type of forums with government as well, with industry, scientists, and regulatory agencies, it's important, right? Because sometimes we have some information, but sometimes we don't have all the information, and I think it's very challenging. I don't think it's impossible, but I think we need to find or to create these forums for discussions, right? Like the one that we have right now here.
Yeah. So I'll share a little bit of a broader view that is being actively debated in the task force I'm on. And it's kind of a combination of quarantine and testing, and saying, at least in the short term, if I wanted to have very high confidence that nobody infected got on the airplane, what could I do? Okay. And so if in mass you had everybody that was going to go on a trip had the requirement to say, okay, you need to pre-quarantine before you go on your trip. So pick the right number of days and actually create a health log of saying, either through my Wi-Fi connection log or my own personal notes, I have not been in contact with more than 5 different people for a 10-day period before my trip. And if I had a test that had a very high accuracy rate, and say, okay, and then I took that test a day or 2 before my trip and I am not infected, then go, that would probably be the best thing you could do. Now there's some risk of, as you're traversing through the airport, you have some non-zero risk, but if everybody had to do that, then you know you're being exposed to other equally secured people, and therefore the odds of it getting worse, the risk is minimized, right? Because even if you filter on the airplane, you put the antiviral materials on the airplane, it's really trying to manage it after you let the cat out of the bag, right? The best thing you can do is don't let somebody who's sick on the airplane in the first place.
Yeah, but I think that I mean, yeah, the social distancing inside the planes, and well, if people are not sick, then that's okay. But if they are sick and they're or asymptomatic, that's so if someone is asymptomatic and was tested negative, then what can it be done, right?
Yeah.
So I hope we can find a way, right? We can detect the virus and also make the virus visible, right?
Yeah.
By naked eye. I think a while ago I saw this kind of publication, but I forgot. Otherwise we have some equipment. For example, Marisha, you talking about use microrama, can detect the virus, for example. That's really cool. I mean, for the RN in the lab, right? Is there any way? Yes, we can trap the virus. At the same time, we can make the virus visible. That would be very useful for industry. I mean, right? I'm talking about from the management. I'm talking about from the virus management and the control point of view.
Yeah, I think I read about ideas about putting color indicators on masks.
Exactly. I think that's the problem.
Put enough dosage, then it will change. Yeah. I think Mustafa has his hand raised.
Oh yeah.
Yeah, so I just from like a broader like community perspective, we usually have this problem of like the hammer looking for the nail and the nail looking for the hammer, and I was wondering whether, you know, for example, a lot of the talks, especially in Dr. Mauricio Turon's and Dr. Wang's talks, resonated a lot of with my own work, but in many cases it takes a pandemic to have some of the material scientists, and maybe that's not necessarily just for the carbon materials, but any kind of material scientist or any kind of you know, physical chemist who can have some skills and that can be brought to bear on you know, virus detection or virus deactivation or enrichment, like in Mauricio's work. So the workshop like this one is an excellent place, but typically beyond that, like when we go to our carbon conference or MRS or APS or the other conferences, what should we do to make these sort of matchmaking more possible and more common, and even beyond the pandemic situation that we're in, and have it more sustainable, where people would, you know, meet people from virology backgrounds, from medical backgrounds, and so on. So do you think from like...
So I mean, right now, I mean, I can tell you from what I'm doing, I mean, recently, right? So now NSF is talking about this convergence, right? So trying to innovate new fields and gathering people from completely different disciplines to work together on a common sort of problem, and having a new field. That means you need, first of all, people need to understand each other, and sometimes that's very difficult because you have different languages. But when you have that problem, that means that maybe that's a real convergence. It's not the same as multidisciplinary research. It's deep convergence means something else, which is really a new field will emerge from this, right? And that's what I mean. I can tell you the experience with the papers we have on this topic. We had promised to publish them. I mean, because the editors didn't find it interesting, or they couldn't find and they couldn't find reviewers, because some reviewers were virologists, some reviewers were, and there was no reviewer who could really assess the whole paper as such. And that's when maybe, yeah, that's a problem to solve. But I think we need to have these sort of forums. But I don't think these societies will. Well, they can help to have this convergence, but I think we need to open up to other disciplines and to open up to to companies as well. But sometimes I think interactions with companies need to be strengthened as well, because they have other ways of thinking, and then they have different timelines compared to what we are doing. And I think but I think identifying the problems and having a common objective would be one way or one step forward. And again, companies know what to do in order if you want to scale up something or you want to produce a device, they know how to do it, but it has to be passed through different tests that we still even don't know, because we don't have a protocol. How can we reach that sensor that will identify viruses in 1 minute? And if you talk to virologists, they will say, well, no, you don't have these gold standards, you don't have that. And then, but there's no protocol, there's nothing that we can say, okay, this is the way to do it, right? So we're facing a real problem that we need to have lots of players and lots of talking, but we have to be effective in the discussions as well. So I'm not sure, maybe I didn't answer your question, but I think that's something that we need to promote.
So
yeah, Yashing and Swami.
Okay, all right. I'm just a millisecond earlier. I would just add to that I think it's very important to study these problems ourselves, so that you have some sort of baseline. And I mean, that's what I was trying to share in my talk. And through the process, my confidence level built up drastically. Because the moment when I talk to virology experts, that I was hoping to learn some smart wisdoms and all things from them, but then I found out that they knew very well how, for example, how this recognition, molecular level recognition works, how you can sort of engineer that. But at the moment that one of them told me that, you know what, the masks are not useful, like even the N95s, because the porosity is way too big, and these virus particles are just like a 100 nanometer nanoparticles, right? Now, at that time, I wasn't even using the argument that there's electrostatic, but just that statement made my confidence level shoot up drastically, because I immediately asked the question back. I said, how often are virus particles released as naked individual nanoparticles? For those of us working with nanoparticles before in the lab, that's what we want in the lab. We want as clean samples as possible, but just spit in your hand and dry it, or spit on a glass slide and look at it on a glass slide. You see tons of other junk. And then I asked the question back, he said, and then it turns out that, not every one of them actually even thought about these, right? Even focusing on the virus themselves. So I realized there's a misconnection. There's like a missing connection right here. I mean, apparently this is not extremely well known. The other thing is that I start to ask the question back. I think maybe this is why these respiratory disease viruses, maybe this is why they are so stubborn, because I think it might be an evolutionary benefit for them. When they are released, they must trigger the release of a bunch of other things to protect them. And I think this is why they can be so stubbornly stable on smooth surfaces. Because if you look at that, it makes no sense. It's a tiny soap bubble. Why is it so hard to break a bubble, right? Why can a soap bubble stay so long in air? So it was, for me, that was really the two transformative moments that I realized that, there's a way that we can actually help. So I would suggest that first we need to talk to them, but I have found it very difficult to talk to clinicians. I have some glorious failure at Northwestern. A few years ago, we organized a whole day outreach with a medical school, and then it turns out that 10 MSc faculty showed up. There's only one person from the entire medical school staying with us, right? So we understand that because they're busy, right? They're saving lives. So that gave me a wake-up call. Next time I approach them, I have to have a very well-defined question. So that's what we did with those doctors in China. Then they were surprised that we seem to be able to at least have a baseline to start with. Otherwise, they'll be like, "Who the hell are you guys wasting my time?" Right? So, I feel very encouraged by that. So that's why I've been telling my students and my colleagues around that maybe just maybe read a few chapters of a biology textbook, start from there, and talk to infectious disease expert. Okay. Or read through all the WHO, CDC general guidelines and try to think about the reasons behind that. Okay.
I think having a common objective is key, and I think that's then you can talk to everyone. Everyone is sort of is excited about solving the problem, right? So.
There's one thing missing that I think is very urgent is that I think Mark also mentioned that for companies they certainly will welcome if you have a new antiviral product. And by the way, the antiviral product coatings, they vary highly based on the application scenario. That you are going to use drastically different composition and release mechanisms. But on top of that, it has been very difficult to actually test these things. So there are universities that can do these things, but for all of those who have access to these viruses, doing such tests is time-consuming, very low-tech, and very low, not very rewarding. So they're very hesitated to do this for you, okay, which is understandable. Now for us, it's also very difficult to even budget these things in our proposals. So I have personally found out this is very, very difficult, and reaching out to people outside the US would actually make it a lot easier, but then you're gonna find a lot other hurdles, okay? So I just wish to see there are more commercial testing services available because there are ASTM and ISO standards specifically for like a textile surfaces, coatings, to certify the antiviral performance. Those are very well designed. We could just tap right into that, but there hasn't been many of those available, or at least very visible. And also they are quite expensive. My entire ONR grant will just be drained by that one test. So yeah, so that's a missing need. I think if we have that connection set up, it will streamline the transition from lab to, let's say, to users. Okay. All right.
Thanks. Yeah, I think having standards protocols it's something that they're not quite there, and especially for something new or like this, right?
Yeah, I'm wondering whether the two comments I want to make. One is I'm wondering whether the initiative that Professor Huang talked about at Northwestern, whether we can use that initiative to develop a framework for how this research should be conducted in terms of defining certain methodologies and some round-robin experiments, and how we can move from laboratory to clinical testing to increase the confidence in the community that this will work or not work. I think that could probably, given the current pandemic and the awareness that it has caused, I think there'll be widespread support, I would imagine, between universities and medical schools and so on. If we can start discussing a common approach to study this, because everybody does their own thing. Even there's no unanimity in terms of how the testing should be done and so on and so forth. So that is one comment. And the second comment is, if we want to attack one problem, let's say out of, I think 3 things were pointed out, how do we contain it? How do we sense it? Then how do we do the antiviral? Based on the literature that I could review, I mean, again, it's not exhaustive. It looks like copper seems to be working so well against many bacteria, many viruses, at least based on the reports, right? So is the community, can it converge on, say, let's say, take copper and see what we can do to make it better, make it sort of a universal antiviral thing for whatever viruses that could come in the future, and be prepared to, even if something is not known, whether we can adapt that copper so quickly to make it an antiviral property. So is that something, an initiative that could also come out of all this discussion? So then focus on something that we know, or at least based on the literature, that it has worked against many diverse bacteria and viruses.
So I would chime in that I would resonate with that last comment about using copper in its current form or characteristics or something expedient and get it deployed. Because a lot of times in industry or these problems, my personal opinion is I hate it when I'm in a situation where I get nothing until I get everything. I would much rather say, copper is a good candidate. It is likely to be effective in a large number of cases. So get it deployed and be doing something so that I'm at least taking out part of the threats.
Right.
And then in the meantime, we can be studying, okay, now how do I beat that? How do I do something better than that now?
Exactly.
That get the rest of it, rather than keep studying it all. And as the time elapse, I continue to effectively doing nothing, even though we are all very busy.
Mm-hmm. Yeah, I've also chiming in that copper, it has been shown to work for even things like influenza. So there have been abundant medical observations on that. And to me, my personal, insight in this is that we don't necessarily need to know a new molecular mechanism of how this works. The difficulty is really about how to do control release. And this is what material science collectively have like a huge collection of knowledge, right? Control release. Because in some scenario, you want a very fast release. You want a coating to be readily reapplied, can be reapplied quickly. In some cases, you do not want to replace a coating at all. So in all these different application scenarios, you will have to formulate your coating or your protective layer differently. That's actually the real difficulty. So there are a lot of these user-centered and applied research design that a lot of us in university are not used to. So we can have something work beautifully in the lab, but it could be totally useless or the wrong direction. So in this part, I think it's very important to have forums like this, and perhaps more forums like this when we actually directly interface with potential users, right? What is your need in this? What is the scenario? For example, for antiviral textile, do you want this to be one-time use, single use, or do you want it to be washable? If it's a single use, it's a lot easier. If you want it to be washable, you also need to worry about, can you reload copper into it? Are you going to release too much copper in the environment? Are you going to poison all the fishes? Are you going to cause allergic reactions? So there are lots of things to consider, but my point is that I think the basic science has been worked out already. We just need the right connection to actually put these together.
Can I just make a
Suresh, yeah.
I think these are excellent points, and I think I concur with Professor Swaminathan. So we need to really understand two aspects here. A virus, when it's inside a cell or animal body or human body, or when it is outside the host. So it's two different beasts we're talking about. So all the molecular mechanisms that we need to really understand, and whenever a new virus comes, what we do in research is really trying to understand what is happening in the body, that is virus-host interactions. So in the context of development of antiviral drugs, which is, we're talking about treating somebody, right, infected with the virus, it is always challenging because one therapy may not translate to the other therapy. So hence we need to really understand what this virus does to the cells and where can I treat it. Now flip the coin, we're talking about inactivating the virus. So technically when it is outside the body, you're not really talking antiviral, but viral inactivation. So, and I think Dr. Swaminathan makes an excellent point, because if you look into all the different viruses that evolved, they essentially belong to a certain families that we already know very well about. And they may have different disease-causing abilities within the members of a family, but if you look at the broad physicochemical properties of these viruses, they are very similar, hence they are part of one family. So it is perfectly possible, if you're talking about copper as a surface that can inactivate a broad range of viruses. So you could test that hypothesis in theory by taking a representative member of every family, since you know this particular material is can inactivate all these different families of viruses, and theoretically can then work as a pan viral inactivation surface. So I think there is a great deal of technological advancements we could do in terms of what we can do to beat the virus when it is outside the body. So, and the question about what happens when it goes into the body and how you can treat vaccine, it's a different beast, and I think that's where we all struggle. But I think that real innovation can be done with people like all of you. We have excellent technological skills in material science engineering, is to come up with a more broad spectrum, generic ways of inactivating the virus when it is outside, on surfaces, in environment. So there are now talks about how do we inactivate the virus in air circulation, for example, or in airplanes. So these are some of the things that we don't really have to reinvent the wheel every single time when a new pandemic happens, because this virus we know from a long time, and there are multiple members of this Coronaviridae family, and most of them behave very similarly. So we have candidates that we can use as surrogates to understand these different families. So I think that is one area where forums like this can make a real difference.
Yeah, thank you. I think that, yeah, I agree. Yeah. So someone from the public or someone would like to say something or has a question or a comment?
I have like comment and question to Suresh. Suresh, when go back to physicochemical properties of virus. So my question is, do we still understand physicochemical properties of this specific virus if you say that line in the same, on the same? So why this virus, specifically this virus, is stable under very high temperature at 85 degrees centigrade? For example, if you compare to regular virus like Spanish virus or regular virus, what is the difference between this virus and regular virus?
Yeah, great question.
So, and this is first. And the second, when you have transmission, regular virus, the strength of the virus is decreases. If you go back to the COVID, SARS-2, transmission is even increases. So depends on the human body. Depends on the human body and immune system. Some patients can die during few hours after COVID. COVID infection. Some patient has under mild conditions. So the doctors right now call problems the kidney pneumonia. So the problem that as during the, so that is you get a lot of infection in the and patient is dying from the kidney. So this is my question.
Okay, so great. You packed a lot of stuff into that question. Perhaps I have to give a lecture, but maybe let me try to keep it simple. So there are multiple aspects you mentioned about the physicochemical properties. So the reason we group viruses into a family because they have a lot of things in common. They have a similar structure, similar proteins, or similar nucleic acid composition. So naturally, you would anticipate that most of the physicochemical properties of viruses in a family are fairly similar, although there will be subtle differences. The second point about your question is, why is this virus is stable in the environment? Why not you call regular virus, I'm guessing other viruses that are currently circulating. So let us take, for example, influenza or flu virus. So the reason this virus is spreading in summer and flu does not do is not because that this virus is extremely resistant to temperature. That's not the case. And the reason is that a virus that circulates in a human population for a certain period of time, what happens is a sizable members of the population are immune. So just think about this. So you have a plain field with all dry grass and a second field where there are certain pockets of dry grass and remaining part of the grass is green. So if you lit fire to that, the fire in a lot that has intermittent dry and green grass will have a very slow spread because it is constantly being stopped by something that is not very combustible. Whereas think about setting fire to a very dry grass and the fire would just sweep through the whole thing. And apply the same analogy here. And the reason that this is spreading even in summer, winter, whatever, is because the virus is constantly finding a susceptible host adjacent to it. So it is transmitting because of people being susceptible. So what would then happen is after some time, maybe come same time next year or in a year's time, a lot of people across the world would have exposed naturally to the virus and developed certain level of immunity. Hence, the virus spread would slow down. So it would then perhaps, it is highly predicted that the virus will then follow potentially a seasonal pattern. And by that time, we call that virus infection as endemic, meaning it continues to circulate or exist in that population. The flu, when it emerges, it's a pandemic, it's a new virus. After some time, it becomes endemic. So that is about that one. And then you talked about, other question was why the virus is killing people or infection kidney, some early, some not. And the other aspect that we need to understand about virus is that the virus by itself do not really cause the death of somebody. So what happens to an individual is how your body reacts to the virus. So if you take randomly 10 people, all 10 people would not react to a given virus at the exact same level. Different people react differently, and it's a very complex biological system that explains why people should react differently. And there are genetic aspects to it, or underlying disease conditions, your age, your race, and a whole host of topics. So now, your body's immune system, when it reacts to the virus, sometimes when your body is seeing a pathogen or a virus for the first time, your body completely gets confused. So it doesn't really know how to react. Sometimes you end up being overreacting. And that's exactly what happens. So virus is infecting different parts of your body, and your body is trying to fight back. In that process, there is an extreme immune reaction to the virus, but that which is really detrimental to your own body. So the whole fluid is getting filled in your lungs, or kidneys are getting completely filled with all these chemicals and proteins. And that will make the function of the organ failed, and that's what contributes to death. So the virus is only the switch, but you are almost reacting to the virus, and that's what causes the. And then the last point, if I remember, you talked about the transmission and then virus becoming gradually less virulent after some time or becoming more transmissible. So what generally happens is when a new virus enters into a new population, in this case, COVID virus emerged from animals into humans, initially the virus, as it replicates, the system is selecting for the fittest virus composition in this environment. So virus is making mistakes, and naturally, the virus particles that had genetic mutations that are better able to transmit get selected, because it's a viral fitness concept. So as it transmits among people, the virus naturally would become more and more transmissible, because we as people, or the environment, is selecting for the fittest composition of virus in that setting. And eventually what would happen is that if majority of the people are either naturally infected and recovered and became immune, meaning they have antibodies, or people have received vaccine, then there is a significant host immune pressure that is driving the virus to change in a certain way, because the virus needs to know, not it has a brain, but if the virus is very virulent and killing the host, it cannot survive. So naturally, the virus host immune pressure would drive the mutation in the virus in such a way that it becomes less virulent, meaning it becomes more less virulent or less severe. With this virus, we are not quite there yet. So the population is not fully immune, and that's what would happen eventually, maybe in 2021 or 22. You can almost predict that this virus will become less and less virulent. And now most of the flu viruses that we know, the fatality rate, meaning how many people would die in a 100 people, it's typically less than 0.1%. And that's what would happen to this virus also. But in order to get there, we still have a long way to go. And one of the major problems is that what is the human cost to that? There will be a lot of people have to get infected, and many people have to die. And eventually we get this so-called herd immunity if we get there. And then that will drive the mutation of the virus into evolve to become more and more what we call as attenuated is the term, because it becomes less virulent or more sober in its ability to cause disease. Hope it answers your question.
Yes, thank you very much. Thank you. I learned something.
Thank you. Sorry, it's a lot of different topics, but I try to keep it simple.
Yeah, thank you very much, Suresh. I think it's great to have you here. So more comments or questions? So I would like to maybe...
Mauricio, I have a question.
Yeah, okay, go ahead, yeah.
I don't know, somebody made a comment about virus immobilization. So what property of the virus, or basically holding the virus in place, so what property of the virus would drive it to actually stick to a material and stay there?
Suresh, do you have...
Again, my take on this question will be from a very virus centric. So I'll try to. So what we know is that the virus, when it is outside a human or an animal, any living body, it is not technically it's not living. It is a particle. So that is one unique thing with the virus, is that that's why we call as virion. So virion is an infectious particle. And when it finds a cell to infect, it becomes the so-called virus, that it has the biological action. But outside the living cell, a virus is nothing but a particle. That's why it's almost like a crystal. So the first discovery in virology is that there is a virus called tobacco mosaic virus. And it was shown by a scientist that a virus can be crystallized, which is kind of fascinating, the thing that how can you even crystallize something biological? And people also argue the fact that viruses are they truly living or non-living? And there is also an argument that they're perhaps in between these two. They are not quite living, but not living. They're in between these two. So, that much I can tell you from the virus standpoint, but what the properties of the surface to attach or stick to any particle would probably also translate into the setting. So I think that is something, perhaps one of the the material science experts need to answer. But think about virus outside the body as a crystal or a particle, and whatever the features that would make this particle to stick to a surface. I'm thinking about the electromagnetic charges or other forces would probably contribute to that.
Yeah, maybe I can add a few comments.
Yes, yeah.
This is one thing I was trying to, I guess, explain in using one or two slides, that when a person releases the virus, respiratory disease viruses, it's always surrounded by other things. There are lipsome-like molecules, there are polymers, there are protein debris, enzymes. We learn from doctors that sometimes there are actually bacteria colonies. There are lots of things. And in a materials term, if you think about it, these are almost like a binders for your battery. So these binds the virus particles together and also to surface. And a lot of these components are also hygroscopic. So they're not really dried out. Okay, so even, I think the local humidity inside that so-called nuclei or dried nuclei, semi-dried nuclei, is actually a lot higher than the environment. So I think that's why they stick well to various surfaces. And this is my personal hypothesis, is that this is why they stay viable for much longer time on smooth surfaces than porous surfaces, because of this local capillary condensation effect, that they can maintain a puddle of water to keep the virus particles hydrated. Because once you dry it up, I mean, it's a soap bubble, right? If we take water away, they should collapse.
Thank you. Thanks.
Thank you, guys.
So maybe I would, maybe if one, the speakers would like to wrap up, and then we can just, some of the ideas that we said, final thoughts, and you can just, with all the discussions we had, what would be the next step, right? So how do you envision having a next set of, if we organize another workshop like this one, what would you like to see, and what would be a next step? I'm trying to do it sooner than later, right? Because we're still in the middle. This is not the beginning of the end of the beginning. And I think we are quite far from this ending.
I would like to ask one more question first. So fomite, contact, and airborne transmissions, and although those are possible, but for viruses, is the distribution typically the same? It's usually via the air or droplets, and it's rarely via surfaces? Or is there something that would flip the table and the next virus could be 85% fomite instead?
So maybe I can try and address that. So perhaps the question is, it depends on, Mark, the virus we are talking about. So I'm taking your question in the context of a respiratory virus. So there are a number of other viruses which are transmitted by many different means. So for example, there are viruses which are insect transmitted. So nothing to do with respiratory droplets or fomites, anything. Insect bites you and feed the blood from you, and then they pick the virus, and when they bite somebody else, they transmit. And there are viruses that are transmitted by sexual transmission, for example. And there are many other different ways. And there are viruses which are gastroenteric. So when people, release the virus through their stools and contaminated water, and when they ingest it, so you are taking the virus in. So there are many different ways in which a virus, it all depends on what virus we are talking about. So let us focus on the respiratory viruses. And there are not many of them. And that is always an absolute nightmare of a virologist. A respiratory virus that can transmit between people is the ultimate nightmare you can think about, because even Ebola is such a deadly virus. We know exactly how it transmits. Unless somebody comes in contact with the bodily secretions of an infected person, there is no way you can pick up the virus from them. So it's much easier to keep the barrier because you know a person is infected or he's sick, and you just stay away from his bodily fluids, whatever, sweat, all different fluids. And if a virus is bloodborne, for example, Zika virus, so all have to be transmitted by an insect vector. Some insect or a mosquito has to bite, so you can protect yourself from a mosquito. Or there are viruses which are sexually transmitted. So, you know, using protection and other measures, you can put the barrier. But none of these things would work for a respiratory virus because this is getting sprayed into the circulation and we do not know the source. So if we clearly know this guy is infected and he's coughing or sneezing, then the individual might be contained. And that's what happened with other more severe versions of respiratory diseases like SARS or MERS. And with this, the problem is we are learning more and more as the time progressed. There are different types of clinical manifestations of people being asymptomatic, symptomatic, and there is this droplet spread. And there is also now evidence coming that there is a combination of droplet as well as maybe a little bit of aerosol transmission. But the aerosol transmission is even more challenging because once it sustainedly happens, then it becomes even more challenging. Now you have the droplets linger in there for some time, they settle down. So you know, if I sanitize my hands, then I can keep a barrier. But if the virus continues to circulate on in aerosols for a longer period of time, there is absolutely nothing we could do because somebody may have sneezed in a room and went away, and I go after half an hour and I still breathe it in. So I think it all depends on the context and what virus we're talking. So maybe SARS, although aerosol transmission is some evidence for it, but it is still significantly droplet infection. So hence, surface decontamination is the most important intervention that we can think about to cut the barrier and the distance. I don't know if I answered your question.
Largely. I was tracking with it all the way up till that last surface is the most effective. Right now in the task force I'm in, since they say it's mostly via droplets, but can be via surface, my position has been the air filtration system and the fact that we filter and recirculate the air every 2 or 3 minutes is the best thing we can do because it pulls all that out, have a very good filter in the system, so then it is now out of the air and the transmit. And that the surfaces, even though social distancing is challenged there, if you have the personal protection equipment or what have you, it is then on the surface, otherwise contained into the seat near you, not the entire airplane. And so I was trying to think of, if we did have very good filtering, and most, yes, respiratory viruses are via droplets, then we could say we believe that we have not only good protection for COVID now, but also for respiratory in the long run, because they typically are via droplets.
Absolutely.
Is that not true? And the next one could be 85% via surface, and therefore all these materials will actually be just as important for the next one that we encounter.
Yeah. I would like to ask something maybe from a non-expert, non-biological expert. I mean, there's I guess, a physical common sense about aerosol is that, yes, aerosol particles, they do stay in air longer. They can travel to a long distance, but if you look at the number volume, right, they are so insignificant compared to other large droplets. So yes, they can go far away, but at the same time, the viruses don't have as a thick protection layer around them. And also the number of viruses in that is also a significant, orders of magnitude smaller. So that's why I agree with Suresh said that. That's why during SARS outbreak, there were a few suspected cases about aerosol transmission, but it was never able to be confirmed. So based on, I guess, so far based on observation, I think droplet is still the majority, accounts for the majority transmission. Aerosol cannot be ruled out, but so far we don't, at least, we don't see this is the majority mechanism of transmission.
And just to add to Mark's comment earlier, so we also must think about where the virus is coming from to be on the surface. So eventually, the droplets will then deposit on the surfaces. The question is that there is just one study which I showed in the slide that tried to simulate the conditions in the lab and said this lives for so long on copper or plastic. But just think about outside. And the virus is, as I was answering one of the questions earlier, it basically has the lipid bilayer around the virus as a covering, which it literally stole from the cell membrane. So anything that is lipid solvents, think about alcohol, soap, anything that can just break the fat, anything that can break the grease is literally killing the virus. So in that way, the length to which the virus can remain viable or infectious on surfaces can be extremely variable. So I don't think there is a real way to model and say, in this condition, this is how long the virus will stay. And that's why I think the surface spread is becoming less and less critical, but more important is the droplets. So when people are near and then the droplets are being, so I think that remains the key challenge. And especially when you're outside in the hot sun, even if the virus gets onto a surface, it probably doesn't live very long because it just gets dried. If you think about even in the lab, if we create a film of virus preparation, if you dry it, the virus is pretty much dead almost instantly. So I completely agree with you that the droplets are a major source. And I think a smart filtration system that can very periodically, frequently can filter out and then pump fresh air can pretty much significantly reduce the risk to a large extent. Although we cannot say it fully takes it away, but you can reduce to a statistically relevant numbers, almost more than 50% or even higher, I think.
But doesn't it make sense in a pandemic, you should really take a hierarchical approach of prevention, droplets, formites, surfaces, etc., right? So it should be a layered approach for pandemic control. If we focus on just only the droplets, then I don't think that's the right thing to do.
Yeah, totally agree. I think the point is that none of these are mutually exclusive, and we must do all of them, as you rightly said. But I think the point is that which is the most significant contributor to the problem? So you really beat the major beast first. So we are not talking about eliminating completely, but how can we have a better mitigation strategy? So what is the biggest problem first? And then there are ways to mitigate. As Mark said, the droplets do play a major role. Can I do something, especially in closed spaces like aircraft or a restaurant room or a classroom? This is what we're talking about. And I think to your point, the other aspects of covering your face or washing your hands and not congregating would continue to remain relevant, and they are not really to be taken lightly, for sure. So all of them are important.
Thank you.
Thank you. So someone would like to wrap up. I think we are basically, this is time for wrapping up. I don't know. Someone would like to.
Maurice,
what shall we do next? What shall we do next?
I've been talking quite a bit, but I just can't stop to share one more thought. Again, perhaps that is also relevant to the next meeting you're talking about. So one of the questions that Mark raised earlier is, say, let us forget about this pandemic for the time being, because we already missed the bus and there is nothing really we can do. Several people are testing and developing tests. Think about how do we prepare for the next pandemic? And one of the challenges in the field is that, let us say if I come up with a very smart device and screen everybody in the airport, and I do find a virus, but then what? What do I know with this? What do I do with this information? So what is the clinical relevance of it? Should I stop every single person that has some virus? I think that's where the key focus and the challenge is. And there are some solutions. For example, now recently we had a discussion about the deep learning and artificial intelligence based models. So for example, if you build, as I mentioned earlier, most of the viruses, whether known or unknown in the future, they mostly fall into the families that we already know. And it's very, very rare that a completely novel virus out of the blue comes up. So one of the ways in which we can build this knowledge is that if we build a a knowledge base or a database that is machine learning based. So the detection can immediately then speak to the system and translate what do we know about this virus, or what is the potential. And some of the companies are talking about this so-called Pangea type approach, where you can immediately make a prediction of, is there anything that is known about this virus that is known to cause disease in humans? So you could make those very quick. So think about a device like what you described, or something of that sort, that can very smartly group the viruses or detect them. And if that feeds into a database or a deep learning, machine learning algorithm that can process the information and say, okay, we are finding this in this individual, what do we know about this? In that way, you can greatly reduce the potential high-risk people that needs further investigation. Most of them that do not meet those criteria can just go. So you can continue the flow of people, the process flow in the airport or other places. So that is one thing that I think a technological innovation can certainly help this rather complicated puzzle we are talking about. So you can proactively screen them, but then still also make sense of what that might mean. And so you have a very, very short list of people that needs to be maybe quarantined or stopped, and perhaps needs a further investigation, and the rest of the people can just go. So maybe that is one area we can think about. It's just an idea for food for thought.
Thank you. I think that's I think again that's convergence research, right? Trying to get different areas working together.
Yeah, maybe a quick mention about this. I mentioned there's this organization that's going to turn into an NGO. It's actually called Advanced Materials Pandemic Task Force, AMPT. Okay, so this is what they're trying to do. They're trying to connect people from different sectors and try to build up a database about what is needed and what are the current capabilities. Try to basically find this match. Now, one thing that almost all of us found is pressing need. A pressing need is to really engage with testing, with a certifying or virus activity testing service. So there are a few companies outside the US that are capable of doing this. I mean, some located in Europe, Singapore, China. In the US, we're trying to discover more of these capabilities around, because they already exist for other viruses. So I think, if we can streamline the interaction, that will work out really well. So I will send that information to Maurizio, and maybe you can share it with everybody. If you guys are interested, feel free to connect with them.
Sure. Yeah, that's very good. Yeah, I think this type of task forces, that's a way to also interact. These are the forums. Yeah.
Okay.
Thanks, Yashing. Wei Ming, do you want to say something?
Yeah, I think probably just to repeat what I said before. I think there seems like a good opportunity to let us work together in the virus detection. I'm talking about fast detection, right, between industry and the academia. Yeah, if you guys find that there is we can write a proposal, for example, we can work together. Yeah.
So I might actually, as editor-in-chief of Carbon, which is maybe that's easier for me. So what I will try to do is I will try to organize, if you guys, if the speakers would agree, trying to have a short perspective, and we can publish it very quickly in Carbon, and trying to maybe just put it in words, the discussion we're having, and also future possible ways to move forward, to not for the current pandemic, but also for the future and things that might happen in the future, right? So I would try to put together something. I will contact you guys because I think it's been a very fruitful discussion, and I think there are important points we need to put out there, and also trying to make more people aware of this.
That's good.
Very well.
Okay, so Wei Ming, do you want to close the event?
Yeah, I think it seems like a long day. We're trying to reduce the time for each and speak. In terms of still a long day, I think we had a lot of good discussion for each the talk in the morning and afternoon, and also I think we have a very good and a long table discussion. We focus on the virus and detection, and also we're trying to see if there are any collaboration opportunity to work together. Like I think like one person said, probably not necessarily just focus on carbon material, right? We need to look at the other and opportunity. So that's from my side. And Arisha.
Well, thank you very much. Thank you, everyone. I think we had a very good attendance to the workshop during the whole day, and thanks for your patience. We were a bit over, I mean, in the talks, but I think the discussions was very good, and I think most of the questions were answered. And this recording will be available for the American Carbon Society members. I think eventually will be put so you will have access. But I will try to draft something and work with some of you guys in order to have something published in Carbon. So I would like to thank everyone, and we'll be in touch.
Thank you.
Thank you very much, Dave, for coordinating all the events.
Okay,
Thank you, everyone. It was a good experience.
And Monica and everyone, yeah, Julian as well.
Thank you.
Thank you, guys.
Thanks.
Bye-bye.
Bye-bye.
Bye-bye.